Hi, my name is Ana, and I've been doing research since the year 2000. What happened in the beginning of the year, or better saying at the end of last year, was that the world changed dramatically. I think we all knew as virologists that viruses are really important, but I think the world was shaken to the ground with these new coronavirus. And we knew that we had to react quickly and divert all our efforts at an institutional level to try to understand this virus as much as we possibly could. And there we were in March with the first case, with the first patient infected in Scotland. So just before that, we had started to kind of reallocate our efforts in preparation for that. In such a way that when the first case hit Scotland, we did very quick characterization of the viral genome. And this was only possible thanks to a fantastic coordination with the diagnostic teams and public health, Scotland, and also ourselves, the academia. And similarly to what happened in the rest of the world, the number of cases started increasing dramatically in Scotland as well. So in terms of the information that we get from these genomes, it's important to understand that they encode all the genetic information of the viruses. And viruses are, they are very simple. And at the very least, they will contain information. They will contain information for the capsid protein, which are pretty much the proteins that protect the genome of the virus. But they will also contain information that encodes for what is called a replicase, which is the protein that enables the virus to do several copies of itself. But most of the virus will contain a bit more of information, like, for example, coronavirus encode for a protein called the spike protein, which exists at the surface of the virus and enables them to interact with the cells. And we know that this interaction is really, really important. So in the case of coronavirus, these proteins at the surface of the cell, they form like a crown. That's why they are called generally coronaviruses. But they also encode many other things like proteins that enable to shut down the defense from the host. So it's not surprising that doing genome research is so important. So how is it that we go around and get this information on the viral genomes? Well, using a technique that is called viral sequencing or genome sequencing, because it can be applied to anything that has a DNA or RNA. And nowadays, these techniques have evolved so much that we can do these very quickly in the lab with some of the devices called sequencers, giving us a turnaround time of just a few hours. And this has been incredibly helpful. But what we also have in the lab are other pieces of robotic equipment that enable us to do large number of samples in one go. And as part of a pandemic response, the high numbers and the rapid turnaround are really important features. What's really extraordinary as part of this outbreak response is that in just a few months, there were thousands of viral genomes available worldwide. So at the moment, there are just over 60,000. And this really demonstrates how this technique has evolved over the last 10 years. What's really interesting is also that the UK has contributed with more than half of those genomes. So we have produced around 38,000 genomes. And that was only possible thanks to concerted effort that is being driven by a consortium called COG UK, which includes not only the NHS, but also public health bodies. But most interestingly, there are more than 12 academic institutes that have been now brought into these consortium. So it just highlights that in terms of the response to this virus, research has been having a strong component, and that's very interesting and reassuring to see. So in terms of applications of this type of techniques, when the first viruses were identified in Wuhan, it was actually genome sequencing that really helped the identification and characterization of this virus as a new coronavirus. But there's more to it than just the characterization of a new virus and also trying to understand the replication cycle of any new virus or older viruses. Because these viruses, every time they replicate, they introduce errors in the new copies. And we call mutations to those errors. But that's really what enables us to trace how the virus spreads and how it transmits. The fact, if the virus was always the same, if it didn't change at all, there was no point in generating 60,000 sequences because they would all be the same. The fact that it mutates, that it evolves, allows really to track how the virus is spreading across the world. And in fact, we decided in Scotland to, well, at the CVR, in collaboration with our colleagues in Edinburgh, that have also been contributed to this sequencing effort, to look at the first month of March and trying to understand what happened to this virus, how did it spread in Scotland. During the first month, there were just over 2,000 cases identified by the test, which is a PCR test. And of those cases, we had initially sequenced around 20%. Nowadays, we have sequenced nearly 66%, so we are reevaluating our initial analysis, but we could see several things. We could see that quite a number, well, there were around 133 introductions at the least, so this is kind of a conservative determination of the number of introductions, and they were mainly coming from Europe. So in order to understand what had happened during that first month, we took not only the virus genome data that we had generated, but equally important, we took the clinical and the epidemiological data set. So we had information on whether the patients had traveled or not, and where did they travel to. And this is really important. It's really putting together all these pieces of information that enables us to draw some conclusions. So during the first 4 weeks, there were at least 113 introductions in Scotland, and they were coming mainly from other European countries. The cases where we had confirmed travel information predated both UK travel restrictions and also most of the restrictions for traveling in other European countries. So we know we could conclude that the emergence of continental Europe as the epicenter of the pandemic was a very important driver of the Scottish outbreaks. And we could see that not only from the travel information, but also because the lineages, the genomes that we were detecting, they were related to European sequences. And what was interesting was that cases with links to China and other countries in Southeast Asia, where the original cases appeared, were comparatively not detected. So like I said, we are now expanding the analysis, and we can drill into this question with a higher depth. But generally speaking, we don't think there's going to be many cases coming directly from China. What we also found out was that some of these inferred introductions were not associated with reported travel. So we could conclude that some of the early introductions of the virus were undetected. And that was when there was a quick establishment of community transmission of viruses in Scotland. So as soon as 3 days after the first detection of infection, we were able to identify viral genomes with no link to travel whatsoever. And this indicates that early introductions to Scotland and community spread occurred before the first case was even detected. Looking ahead, we've started analyzing the data already to try to understand the effect of the lockdown in terms of the spread of the virus in Scotland.